Let me check another question. What disorder? What is the mistake? Okay. Let me speak about Shigella, one of the other important members of Enterobacteriaceae. This is like Salmonella, very pathogen, very virulent, and infection dose of this bacteria is low. It means the small number of bacteria can enter to your body and make you disease. Taxonomy of Shigella is based on O antigen. O antigen, LPS. When we speak about O antigen, we remember LPS, lipopolysaccharide of cell wall of gram-negative bacteria. Based on LPS and some biochemical reaction, we classify Shigella in 4 groups: A, B, C, D, or 1, 2, 3, 4. Shigella dysentery, the most serious form of bacteria, the most important, the most pathogen, the most virulent of bacteria, Shigella dysentery. 2 or B, Shigella flexneri, the cause of shigellosis in underdeveloped countries. Number 3, Shigella sonnei, the cause of shigellosis in developed countries. And Shigella boydii, the number 4. We have 4 categories, 4 classes of bacteria which are classified based on O antigen. As the other members of Enterobacteriaceae, they are gram-negative, facultative, anaerobics, coliform bacilli, non-motile. They don't have H antigen. Process capsule, they have K antigen, but classification doesn't do based on K antigen because it confer or interfere with O antigen. And they non-lactose fermenting, it means on selective media like McConkey agar, they do not make colorful or pink colonies. They boil salt resistant. It means they can grow on McConkey agar. You know, in McConkey agar, there are boiled salt. So just the bacteria which are boiled salt resistant can grow. But colorless bacteria, they make colorless bacteria because they can't ferment lactose. Like the other members of Enterobacteriaceae, they are glucose fermenter, they reduce nitrates, and they are oxidase negative. But now we want to speak about pathogenicity of this bacteria. Shigella causes disease by invading. Shigella, like Salmonella, is an invasive bacteria. So by invading and replicating in cells, structural gene proteins mediated the adherence of the organisms to the cells as well as their invasion, intracellular replication, and cell-to-cell spread. These genes are carried on a large virulence plasmid, but regulation by chromosomal gene. What does it mean? The gene for these virulence factor are on a plasmid, but regulation of genes is done by a chromosome. Shigella species appear unable to attach to differentiated mucosal cells. Rather, they first attach to and invade the M cells located to pear patch. Wait a moment. You can see here, but again, at first attach to M cell and then invade M cell which are located in pear patch. You can see invade M cell and then lyses the vacuole, exist out of the vacuole and replicate in the cytoplasm. The type 3 secretion system mediates secretion of 4 proteins. The type 3 secretion system is very important, a Byron's factor. They mediate secretion or delivering of 4 proteins, IPA-A, IPA-B, IPA-C, and D, into epithelial cells and macrophages. These proteins induce, and these are the ability or responsibility of these proteins. 1, membrane ruffling on the target cell, which lead to engulfment of bacteria. And then Shigella lies the phagocytic vacuole and exit out of it and replicate in the host cell cytoplasm. Unlike Salmonella, as you observed in the last slide, which replicate in the vacuole. With the rearrangement of actin filaments in the host cell, the bacteria are propelled through the cytoplasm to the adjacent cell, in which cell-to-cell passage occurs. In this way, Shigella organisms are protected from immune-mediated clearance. You know, Shigella can migrate from one cell to another cell, from two neighbor cells without existing from host cell and face to the immune cell. This is very good for it because they do not face to the immune cell and protect itself. Shigella survive phagocytosis. How? By inducing apoptosis. This process also leads to the release of some kind of interleukin called interleukin number 1 beta, resulting in the attraction of polymorphonuclear leukocytes into the infected tissues. This leads to the destabilization, the integrity of the intestinal wall. Shigella dysentery produce an exotoxin. Besides the other factors, Shigella dysentery produce an exotoxin called shigotoxin. This is similar to shigotoxin produced by enterohemorrhagic E. coli. We spoke about it last session. This toxin has 1 A subunit and 5 B subunit. Wait a moment. Yes, you can see here A subunit and B subunit. And B subunit, we have 5 identical or similar B subunit. The B subunit binds to a host cell glycolipid GB number 2. GB number 3 is a receptor which you can find it on epithelial cell, in gastrointestinal tract, and on glomerular endothelial cell in the kidney. And after attaching to the glycolipid, the number 3 facilitate transfer of A subunit into the cell. The A subunit cleaves the 28S RNA in the 60S ribosomal subunit, preventing the binding of amino acyl transferase RNA, and as last or finally disrupting protein synthesis, disrupting or stopping protein synthesis. The primary manifestation of toxin activity is damage to the intestinal epithelium. You can see this toxin has two parts, two subunit, A subunit and B subunit. B subunit include 5 identical parts, while subunit B attaching is occurred. It is attached to GB3, and then A subunit enter to the cell, and protein synthesis is stopped. So host cell is damaged. Host cell is damaged. And as I told, GB3 is found on the glomerular endothelial cell in the kidney, so shigotoxin can mediate damage to the kidney, HUS. Unlike to Salmonella, Shigella just has human as its reservoir. Humans are the only source for Shigella. We have no animal as source for Shigella. It is proved, it is estimated that almost 500,000 cases of Shigella infections annually in United States, and among different strain of Shigella, Shigella dysentery is more dangerous, and after that, Shigella sonnei is responsible for almost 85% of infections, and flexenary predominant in developing countries. Shigellosis in general is primarily a pediatric disease, with 60% of all infections occurring in children younger than 10 years. Children younger than 10 years. Endemic disease in adults is common in male. Endemic disease is in adults, in male, homosexual, and in household contact of infected disease. Shigellosis is transmitted person to person by fecal-oral routes, primarily by people with contaminated hands and less commonly in water or food. Because as few as 100 to 200 bacteria can establish disease, shigellosis spreads rapidly in communities in which sanitary standards and the level of personal hygiene are low. It's very important. And the symptom of shigellosis, abdominal cramps, diarrhea, fever, and bloody stools. We have 1 to 3 days incubation period, and after that the symptom is started, and by elaboration of enterotoxin, the symptom, yes, you can see the symptom. But the good point of one point again. When you're sampling a fetus, you can find the neutrophil, erythrocyte, mucus on the stool. But one good point about shigellosis, it is generally self-limited. What does it mean, self-limited? Self-limited means this problem, this disease is controlled itself, and the immune system can control this disease, and it doesn't need to treat yourself. Or although antibiotic treatment is recommended, why? To reduce the risk of secondary spread to family members and other contacts. A symptomatic colonization of the organism in the colon develops in a small number of patients and represents a persistent reservoir for infection. So, okay. So, self-limited or self-controlling of this disease of shigellosis is very important, and we can say it is a good point of this disease, because for some cases, of course, you should consume antibiotic, and you should, of course, receive some important treatments. But about some digestive disease, self-controlling, or your immune system help you to control your health situation. Do you have any problem, any question about Shigella or Shigellosis? Not entire the digestive system. No. As I told, wait a minute. As I told, Shigella can attach to M cells, which are located in pear patch in a small intestine, and colon. Because glycolipid GB3 is can found in colon. Yes, just a small intestine and large intestine colon, not entire digestive system. Okay. Just the best way to prevention of shigellosis. Shigellosis means infection due to Shigella, or salmonellosis, infection due to salmonella. The best way to prevent of shigellosis is increasing the level of hygiene, increasing the standard of hygiene. You know, as I told in two sessions, the some disease related to digestive system, some gastroenterite, are seen in developing countries, are seen in summer in some regions that the level of hygiene standard is low. So the best way is to increase the level of standard hygiene standard level, washing your hands, don't eat uncooked food, don't eat food in some restaurants that the level of hygiene is low, don't eat salad in summer in restaurants, and consider some hygienic matters to yourself, some standard material, some detergents in your work office, in your home, some for example, something like this. You should consider something. You are microbiologist, and you should more attention to something like this, because these bacteria are transmitted by oral fecal. So you should consider what you eat, what you should consider the water you drinking, the food you eating, so you can prevent it easily. Why don't you choose Salmonella cytoplasm for reproduction? Salmonella prefer to remain in vacuole, because if we exit from the vacuole and enter to the cytoplasm, in the cytoplasm, the environment of cytoplasm is not suitable for replication, and in the the protease and the other enzyme in the cytoplasm is not suitable for it, and it is killed by them. So it remains in the vacuole, and in the vacuole, the condition is more suitable for it to replicate. But for Shigella, the environment of cytoplasm is more suitable for replication. How to therapy Shigella infection? Therapy, as I told, Shigellosis is self-limited. It means your immune system can control Shigellosis, can therapy yourself. You don't need any therapy. But antibiotic treatment is recommended. Why? To reduce the risk of spread to family members. When you are diseased, when you have shigellosis, the risk of spreading, the risk of transmission of bacteria to the others member of family, to your sisters, brothers, parents, is high. For preventing this transmission, it's better to consume antibiotics. So the best way for you is to consume antibiotic for reducing the risk of spreading, reducing the risk of transferring the bacteria to the other members of your family. But if you don't consume antibiotic, after a while, this disease is controlled itself, and there is no problem generally. There is no problem generally, and you should receive some electrolyte and water as serum, because you lose some water. One of the symptoms of this disease is diarrhea, and you lose water and some electrolytes, chloride, sodium. So it's better to receive serum. Okay. Now we want to speak about another members of the family of Enterobacteria, which is called Yersinia. I think most of you know this bacteria as black, as the cause of black death. Black death or magnesia, and the etiologic agent of black death, which is cause of 3 main pandemic in the world. It has a long history and long story in all the books of microbiology, and and we want to speak about a brief of this bacteria. Members of, yes, I will send you in a group, of course. The 3 main members of these genus is Yersinia enterocolitica, Yersinia tuberculosis, and of course, Yersinia pestis. As you see in this picture, this bacteria, as the other members of Enterobacteria, is gram-negative, a small rolled shape. Gram-negative, a small rolled shape. A common characteristic of pathogenic Yersinia species is their ability to resist, please pay attention, resist phagocytic killing. One of the most important strategy of your immune system is phagocytosis. By phagocytic cells, you kill all bacteria and viruses, but these bacteria are resistant to phagocytic cells. The type 3 secretion system mediates this property. On contact with phagocytic cell, it means when a bacteria is in contact, is in beside, is in face-to-face with macrophage, the bacteria secrete proteins into the macrophage that dephosphorylate several proteins required for phagocytosis. A phagocytic cell like macrophage have some proteins which are needed which are required for phagocytosis. But these bacteria produce and deliver some proteins by type 3 secretion system, which dephosphorylate these proteins and stop phagocytosis. For example, YOPH gene product induce cytotoxicity by disrupting actin filaments. YOPE gene product and initiate apoptosis in macrophage. YOPJP gene product. Let me tell again. On contact with phagocytic cell like macrophage, the bacteria secrete proteins into the phagocytic cell that dephosphorylate several proteins required for phagocytosis, like YOPH gene product, which induce cytotoxicity by disrupting actin filaments, YOPE gene product, and initiate apoptosis in macrophage. Type 3 secretion system also suppress cytokine production. It's very important. Cytokine production, because by production of cytokine, the other members of immune system come to this point and help to each other to stop or to lyse the bacteria. By suppress cytokine production, in turn diminish the inflammatory immune response to infection. Yersinia pestis has two plasmids. It is very important. Has two plasmids that encode virulence genes. Number 1, fraction 1 or F1 gene, which codes for an antiphagocytic protein capsule, and number 2, plasminogen activator PLA protease gene, which degrades complement component C3b and C5a, which preventing opsonization and phagocytic migration, respectively. The PLA gene also degrades fibrin clots, permitting Yersinia pestis to spread rapidly. Other virulence factors specifically associated with Yersinia pestis are serum resistance and the ability of the organism to absorb organic iron as a result of a siderophore independent mechanism. This is very, very important, and many scientists work on Yersinia pestis to understand these mechanisms, because Yersinia pestis is very pathogen and cause many deaths in the world. So these two plasmids are important. Plasmid number 1, fraction 1, and plasmid number 2, plasminogen activator. Protease gene degrade complement component. The material of this component is proteins. All Yersinia infections are zoonotic, something between animal and let me see in this picture and again back to the slide. Zoonotic, you can see a rat here, some flea here, and the other cat here, rodents. You can see, and human here is accidental host. Human here is an accidental host. In fact, the main reservoir, the main source of the bacteria are animals, and the vector are insect, flea here. But human is an accidental host. Let me come back. Okay. So we can say all Yersinia infections are zoonotic. With humans, the accidental host. There are two forms of Yersinia pestis infection: urban plague, for which rats are natural reservoir, and sylvatic plague, which are rabbits, rats, domestic cats, pigs, rodents, are natural reservoir. Plague caused by Yersinia pestis was one of the most, please pay attention, one of the most devastating diseases in history. And epidemic of the plague were recorded in the Old Testament. And perhaps the first of three major pandemic urban plague started in Egypt. Urban plague is maintained in rat population and is spread among rats or between rats and human by infected fleas as vector. Fleas become infected during a blood meal from a bacteriemic rat. When a bacteria enter to the blood of a rat, and when a flea is biting a rat, bacteria enter to the blood of a flea. Digestive system of a flea. After the bacteria replicate in the flea gut, the organisms can be transferred to another rodent or to the human. Urban has been eliminated from most community by effective control of rats or better hygiene. Yersinia pestis produce a fatal infections in the animal reservoir, so cyclic patterns of human disease occur as a number of infected reservoir hosts increases or decreases. But something about Yersinia enterocolitica, as you see here, Yersinia enterocolitica. Yersinia enterocolitica is a common cause of enterocolitis in Scandinavia. And most studies shows that infections are more common during the cold months. And virulence with this organism is associated with a specific serogroup, with most common serogroups found in Europe, Africa, Canada, and O3 and O9, and in United States, O8 are seen. Yes. And Yersinia pseudotuberculosis is relatively uncommon. It's not common, it's uncommon cause of human disease. But something about plague. We told plague is very important, is a devastating fatal disease, and we have three main pandemic plague in all of the world. we have two kinds of plague. One of them is bubonic plague and the other one is pneumonic plague. Bubonic plague is characterized by an incubation period of no more than 7 days. Please pay attention to me. 7 days after a person has been bitten by an infected flea, patients have a high fever and a painful bubble. Painful bubble, what does it mean? Inflammatory swelling of the lymph nodes. Bacteria develops rapidly if patients are not treated, and the rate of mortality is high, 75%. But pneumonic plague, the incubation period is shorter, just 2 or 3 days, and fever, malaria, pulmonary signs are seen within 1 day, and the mortality rate is very high, 90%. In untreated patients is very high. And 2/3 of all Yersinia enterocolitic infections, we can see enterocolite as the name implies, enterocolite. And the gastroenterite is typically associated with ingestion of contaminated food and water. After incubation period of 1 to 10 days, the patient experiences disease by diarrhea, fever, abdominal pain, and lasts as long as 1 to 2 weeks. And the last is Yersinia pseudotuberculosis, produce an enteric disease, which, as I told, these are uncommon. Yes. And another point about Yersinia is this bacteria grow at 4 degrees centigrade degree, and we can have cold enrichment about this bacteria. I mean, we can, after sampling a specimen, we can put it in refrigerator for 24 or 48 hours. After that, we can culture it, and it helps us to enrichment our bacteria. So this content was about Yersinia, one of the most pathogen of bacteria which cause many important pandemic in the world. The main etiologic cause of Black Death, and just it. Do you have any question about Yersinia, especially Yersinia pestis? Okay. In this family, we have another bacteria that I don't want to speak about them. It doesn't mean they are not important, no. But we do not have enough time. We are going to speak about some bacteria in some families, and so we have to ignore some of them. For example, in this family, we have Klebsiella, and about Klebsiella, all of you know, of course, know. Klebsiella pneumonia, the main cause of pneumonia. You have, I'm sure you see in the laboratory the appearance of mucoid of these bacteria. I put the slide here just for memorizing you, but it's not in your exam. You can see the appearance of it, the mucoid of these colony, because they produce carbohydrate capsule. And on blood agar, you can see this appearance, or on the McConkey agar, because the Klebsiella is lactose fermenter. By fermenting lactose, it produces pink cloning on selective media, like McConkey agar. Or in this family, we have protos, and in this genus, of course, Protos vulgaris and Protos mirabilis, and one of the most features of this bacteria is a special kind of motility called swarming. You can see here on blood agar swarming. Swarming is described as the formation of concentric zone of, you can see here, growth of bacteria like this. Kind of motility is special just for this bacteria, and it is causal UTI, ureteric tract infection, especially in patients who are in hospital. And this bacteria is Ubrous positive. And the other bacteria, Citrobacter, Enterobacter, these are in this family. But as I told, we do not have enough time to speak about all of them with details. So we have to just speak about some genus of each family. So we finish this family here, and from next session we want to start another family of gram-negative bacteria. And I hope you studied the book, and if you have any question, any problem with this matter, please contact with me in the group, or speak in the meeting in the class to the other students. Participate in our discussion and learn more. If you have any question, we will finish the class. Please put me a message in your group to send you this slide. Now one of you put a message in your group. In the event that a person is infected, does it not become contagious, or only the source of the infection is enemy? What? Yersinia, plum, if it is gastroenteritis disease, no, it's not contagious. Because, for example, by eating some food or water, some contaminated food or water, it became disease, and it cannot transfer her or his disease to the others. But if you mean digestive disease or gastroenteritis, no, it cannot transfer